Okay, we're going to get started with the awards ceremony. It's a great privilege and honor for me to be able to welcome the Gruber Foundation and Patricia Gruber. The presentation of the awards are going to be this morning, and the Gruber lecture is going to be this afternoon at 1 p.m. So again, with great pleasure, I'm now going to introduce Patricia Gruber.
Thank you, Neil Risch, for that warm welcome. Welcome today to the presentation of the 15th Annual Genetics Prize, honoring a leading scientist or group of scientists for groundbreaking contributions to any realm of genetics research. We are delighted to be here in Baltimore to present the prize at the 65th Annual Meeting of the American Society of Human Genetics. I'd like to acknowledge the founding vision and leadership of my husband, Peter Gruber, in establishing these prizes, as well as his passing. Peter died last fall after leading a long and productive life. Many know his story. He was born in Budapest, and after fleeing Hungary during World War II, his family settled in India, where he was educated by Jesuits in the Himalayas. Later, he came to the US and went to work on Wall Street. His success as an asset manager allowed us to establish the Gruber Foundation and create the prize program a few years later. To further continue the legacy, an agreement in 2011 with Yale University established the foundation there, where science prizes and human rights programs will continue to flourish. Today, we're here to honor the achievements of Emmanuelle Charpentier and Jennifer Doudna for their joint creation of a revolutionary gene editing technology known as CRISPR-Cas9. Please let me tell you a little about the company they are keeping with our other prizes. The Foundation's prize program, which was established in 2000, presents 3 annual $500,000 prizes in the fields of cosmology, genetics, and neuroscience. Each prize recognizes achievements and discoveries that produce fundamental shifts in human knowledge and culture. The General Assembly of the International Astronomical Union met in Honolulu, and the Cosmology Prize was presented to John Carlston, Jeremiah Ostreicher, and Lyman Page. The Neuroscience Prize will be presented at the annual meeting of the Society for Neuroscience in Chicago to Carla Schatz and Michael Greenberg. Returning now to genetics, we are proud of the Foundation's illustrious laureate roster, and we're very pleased to be adding to it today. This year's prize recipients were chosen by a distinguished advisory board. We deeply appreciate the knowledge, the commitment, and the enthusiasm these advisors bring to the judging process. We would also like to thank the American Society of Human Genetics and the Genetics Society of America for their continuing and fundamental support of the Genetics Prize and the Rosalind Franklin Young Investigator Award. The latter is presented to honor the groundbreaking contributions of Dr. Rosalind Franklin and to inspire and support new generations of women in the field of genetics. We are pleased to announce and congratulate the 2 2016 Rosalind Franklin Award recipients, Maria Barna of Stanford University and Carolyn McBride of Princeton University. Now I'd like to introduce the chair of the Genetics Prize Selection Committee, Huda Zogby, who will read the official citation and present the scientific accomplishments of the recipients. Professor Zogby?
Good morning, everyone. The Gruber Foundation proudly presents the 2015 Genetics Prize to Emmanuelle Charpentier and Jennifer Doudna for establishing a framework for universal genome editing. Charpentier and Doudna discover that the bacterial enzyme Cas9 is an endonuclease that cuts DNA at sites specified by a single RNA and define biochemically the components required for this reaction. They showed that the sequence of the guide RNA could be modified to target the endonuclease to virtually any site. This provided the mechanism by which bacteria acquire immunity to specific viral infections, allowed introduction of specific mutations at desired sites, and provided the means to transfer efficient Cas9-directed break, repair, and editing to any cell type. This method has broadly enabled genome editing for uses in basic biology, medicine, biotechnology, and agriculture. The course of science in the 20th century was fundamentally altered by the discovery of DNA. It appears that the 21st century is going to be fundamentally altered by RNA. So many of you already know and use CRISPR that the system hardly needs an introduction. But the 30-year history is still not well known, and it helps us to appreciate the achievement of Charpentier and Doudna. The CRISPR story began in the 1980s when bacterial geneticists noticed genomic regions of repeated sequences that were interrupted by blocks of random spacer DNA. No one had any idea what this curious arrangement could mean. These sequences got their name as clustered, regularly interspersed short palindromic repeats, CRISPR for short, in the 90s. They were always located near cluster of genes. This CRISPR-associated Cas genes encoded enzymes that cut the DNA, but no one knew why, or what use microbes would have for these repeated sequences. By 2005, researchers realized that the genetic material in the CRISPR spacers looked an awful lot like viral DNA and suspected that the bacteria were using CRISPR as an adaptive immune system against viral infection. The idea was that the bacterium would use its Cas enzymes to clip fragments from the DNA of an invading virus and insert them into its own CRISPR sequence, forming a molecular Rolodex so that it can recognize the virus at the next encounter. But how does this process work? This is where our Ds make a huge difference. In 2011, Emmanuelle Charpentier dissected components of the system and revealed that it trans-encoded small RNA with complementarity to the guide CRISPR RNA precursor, a host ribonuclease 3, and CSN1, now known as Cas9, are essential for maturation of the CRISPR RNA and bacteria immunity. Jennifer Doudna and colleague Eva Nogales crystallized the Cas proteins and showed that they operate like assassins in a spy movie. As a microbe encounters viruses, it collects viral DNA in its CRISPR regions and copies the genetic material in each spacer into a strand of RNA, which is taken by the Cas enzymes. The Cas molecule then circulates through the cell, carrying this piece of RNA, searching for genetic material that matches its viral calling card. When it finds such material, the RNA latches onto it, and the Cas enzymes cleave the viral DNA to prevent the virus from replicating. Realizing that this killer precision could be useful in the lab, Doudna and Charpentier collaborated and delved deeper. In 2012, they discovered that the protein Cas9 relies on 2 short RNA guide sequences to recognize and cleave foreign DNA. They figured out how to provide Cas9 with an RNA sequence that matched a sequence of DNA they wanted to replace with a new sequence. They then modified the system so that it can be guided by a single short RNA molecule. The Cas9 CRISPR system has become the most specific tool we have for editing the genome, and it also makes it possible to edit genes in organisms that have been otherwise difficult to study in the lab. The story of CRISPR reminds us that an observation that seems trivial may become recognizably important only many years later, thanks to the curiosity and creativity of the 2 scientists we honor today with the Gruber Prize. Emmanuelle Charpentier and Jennifer Doudna, please come forward to receive your prize.